Hey guys, so this video is a review of what we have focused on within class surrounding photosynthesis, and will hopefully get you guys to review and rethink the process of photosynthesis. Please take your post-quiz and pre-quiz before and after this so we can measure your improvement. So to get started, let's remember where photosynthesis is occurring. So we have our plant here, and in our tree we have organelles that are called chloroplasts, which is the site of photosynthesis in a plant. And inside these chloroplasts, we have the inner membrane and the outer membrane. And then we also have thylakoids, which is just one single one, although they are stacked into grania, and the stroma, which is this inside area. Now, inside the thylakoids, we also have the lumen, and the membrane of the thylakoid is where our first process, the light-dependent reactions, is occurring. So we have sun going into our light-dependent reactions, which is why it's called light-dependent or light reactions, as well as H2O. One of the products of the light-dependent reactions is O2, ATP, and NADPH. Now ATP and NADPH head on to our Calvin cycle, which is the second stage of photosynthesis that is occurring in the stroma of our plant. Oops, forgot to label that one. Our next process is the Calvin cycle. Remember, this occurs on the outside of the thylakoid membrane in the stroma. I have another diagram here of the Calvin cycle. Pause this video now and label the inputs, outputs, and stages of this cycle. Keep a running list of the inputs and outputs, and feel free to put notes by each of those so you can identify where the input and output is coming from and its purpose in the cycle. Please pause this video now. Again, our first reaction is the light-dependent reaction. On this diagram, I have shown photosystem 2, photosystem 1, the electron transport chains, and ATP synthase. Now pause this video now and do your best to fill in this diagram. Include the hydrogen atoms and its concentration gradients, label the parts of the thylakoid membrane and the lumen and stroma included in this diagram, and any other molecules that carry electrons or energy to this process. Try to keep a running tab of the inputs and outputs of this process as well. Do your best to be as thorough as needed to show what is happening. Some questions to think about when you're going through this are: what would happen if photosystem 1 was unable to work properly? How about photosystem 2? What if those electrons could not move from the photosystem 1 or 2 to the electron transport chain? Or what would be a drawback if we had fewer H2O molecules coming in? Alright. So now let's compare your diagram to mine. What are you missing? Is your flow of electrons and hydrogen similar to mine? Let's go through this. So for our anatomy of the thylakoid, we have our stroma on the outside here, and we have our lumen on the inside, and this process again is occurring on the thylakoid membrane. We have our photosystem 2, P680, and photosystem 1, P700. We have sunlight coming in, and it's exciting these pigment molecules, which excite the electrons, and H2O comes in, drops off 2 electrons, breaks down into O2, which is released as a byproduct. The electrons then flow through the electron transport chain. The energy of this is pushing the H+ hydrogens to flow in from the stroma to the lumen. Now this is going from a low concentration to a high concentration, so the electrons are providing that energy for the movement. Then we move on to P700. Electrons from P680 are still flowing in there. However, we have more light coming in to excite more pigment molecules and more electrons to move on to the next electron transport chain. Here we have NADP+ that is coming in from the Calvin cycle to pick up the 2 electrons coming off here to form NADPH. And that later goes on to our next process, the Calvin cycle. The hydrogen gradient that has been created here adds pressure to ATP synthase to start moving. So this starts circulating, and as this circulates, H+ is pushed through and ATP is formed. Now this ATP that was formed in our light-dependent reactions also travels with NADPH to the Calvin cycle. So the ATP is our energy carrier and NADPH is our electron carrier. Alright, so one question I want you to think about this is, what would happen if we had a decrease in NADPH coming from the light reactions to the carbon fixation process. Let's go through this. Compare your diagram to mine. What are you missing? Is your flow of electrons similar to mine? Do you have similar inputs and outputs? Have you labeled the processes on each step? So the first step occurs, it's carbon fixation. CO2 diffuses into the stroma from the surrounding cytosol. An enzyme combines a CO2 molecule with a 5-carbon carbohydrate called RUBP. The product is a 6-carbon molecule that splits immediately into a pair of 3-carbon molecules known as PGA. Process 2 is reduction. So this is where PGA is converted into another 3-carbon molecule called PGAL in a 2-part process. So the first part is each PGA molecule receives a phosphate group from a molecule of ATP. The resulting compound then receives a proton from NADPH and releases a phosphate group, producing PGAL. These reactions produce ADP and NADP+. These 3 products can be used again in light reactions. So the third step of our Calvin cycle is regeneration of RUBP. So this is where PGAL is converted back to RUBP. This is necessary to keep the Calvin cycle going. Some PGAL leave the Calvin cycle and can be used by a plant cell to make other organic compounds, including amino acids, lipids, and carbs. Among the carbs are the monosaccharides glucose, fructose, and disaccharide sucrose. This is where our plant makes sugar. Other GAL that is left in the cycle is then regenerated. So like I said, some PGAL molecules reform the 5-carbon sugar with the help of energy from ATP. Each 5-carbon RUBP then is ready to enter the cycle again. Do a final check to see if your diagram looks similar to mine. If not, go back and reread your notes or reread your book. Ask us if you have questions. So let's regroup and go through this process once more together. What's the big picture? Why are we learning this? Plants are taking in CO2 and producing oxygen for the environment. We also have plants that are producing glucose from this process so that the plant has food, or sugar. Our first process was the light reactions, which took in water and sunlight. That is where we got our oxygen from, that is released into the environment from the plant. Our ATP, which was also produced from this process, and our NADPH, is then transferred to the Calvin cycle, where it gets used up and eventually returns back. Our Calvin cycle is where our CO2 is taken in. And overall, our process, our big production from Calvin cycle is glucose that the plant can use as food. I hope you all have a better understanding of photosynthesis now. Do not hesitate to contact us for any further questions you have about your exam. You can also come to one of our study sessions and get some more preparation. I wish you good luck, and feel free to also look at the attached videos I have that go a little bit more in depth in this process. Good luck!